All right, so this is going to be the first part of a three-part lecture series on food allergies, drug allergies, and anaphylaxis. So the slideshow file, will be all three of them together, but I'm going to split them up into three different areas with their own concept checks, their own notes, and whatnot. So let's get into it. This is going to be an overview of all of the three sections. Feel free to pause. But the first two are going to be very similar, just talking about epidemiology, their mechanisms, and how they're different, and then some diagnostic tools as well. So as far as epidemiology goes, these are most common in children, and that makes sense because a lot of people tend to outgrow their food allergies. And even though you can develop food allergies later on, we're going to see higher rates of people outgrowing them. So that's why they're more common in childhood. As with many atopic conditions, the prevalence has been rising, and we've talked a lot about how these industrialization factors, globalization, hygienic theory, even microbiotic theory, how all of these influences are causing a rise in prevalence of atopic diseases, especially food allergies in certain areas of the world. But it's really that sort of, distribution, that geographic distribution, that is really interesting. So, for example, if we look at the UK versus Israel, two very different places of the world, we see that peanut allergies are 10 times higher in the United States. So the question is, why do we have lower peanut allergies in Israel? And it's these sort of epidemiological trends that cause us to, were one of the ways that we actually started researching what would eventually become the LEAP study, or the Learning Early About Peanut Allergy study, which basically said that if you introduce peanuts early, which is a cultural habit in Israel, you see lower rates of food allergy prevalence, or I should say peanut allergy prevalence. So this is one of the ways I think it's really interesting to think about epidemiology, because this can really inform the sorts of questions we ask about what research to do and what is causing these things at the most fundamental level. So moving on, there are some allergies that are more often outgrown than others. So things like tree nuts and peanuts and seafood, I have all three of those. Those persist into adulthood. But some of the other ones, like milk, egg, soybean, and wheat, are much more often going to be outgrown, and of those I only have one. And if you're wondering why, well, there is a pretty strong theory behind this, or a pretty strong sort of argument for why certain ones are outgrown compared to others, and we'll get to that in a bit. So something I want to talk about is just the genetic risk factor. So this is not something that is often the center point of food allergies, but it is relevant. And we see that if we look at twins, for example, there seems to be some genetic contribution, because there is, in one study they found around a 64% concordance of food allergies between those two twins. So again, that suggests that we should be looking at genetic factors. And one of those factors that we identified through a GWAS, or a genome-wide association scan, found this MC locus as a potential risk locus. And what this MC locus contains is a bunch of genes that have histone proteins, or histone-related proteins, that are involved in epigenetic regulation. So what does it tell us? Well, that tells us that epigenetics is about how our genes are modified by our circumstances. So again, this is actually, even though this is sort of in the field of genetics, this lends a lot of credence to the theory that the environments we grow up in are really influential in our development, or our resistance to food allergies. We also see that this MC locus is associated with many other atopic conditions, one of which is EOE. It's also associated with being sensitized to multiple allergens. It's associated with Crohn's and other sort of immune diseases, which suggests not even just a specific atopic function, but a general immune dysfunction role in our body. So let's talk a little bit about some of these other risk factors. So we talked about some of the more endogenous ones, but things like being a C-section baby tend to increase your risk. You don't get those bacterial flora that you typically get when you pass through the birth canal, and that is something that is sort of starting to change. Gut dysbiosis. So we see that certain levels of certain bacteria are more associated with potentially resolving, in this case, a milk allergy. So that's interesting to think about. And this is one of the most exciting places about food allergy research. Um, we see that vitamin D in sunlight tends to play a role. So we have more allergies in regions that are, colder with less sunlight. And if we see that mothers who take vitamin D supplements tend to lower the risk of having their baby sensitized to food. So again, that's very interesting to think about. And another thing is in more Western diets, we tend to have a really high sort of count of omega-6 polyunsaturated fatty acids. And these tend to be pro-inflammatory and lead to levels of, prostaglandin E2, which is, of course, a very big player in allergic inflammation. And we tend to have less of the N3 polyunsaturated fatty acids. So this is one of the ways that diet and that combines with vitamin D. So all of these things sort of gesture to the way that we take care of ourselves as humans. The environment we grow up in is going to have a really big impact in food allergies specifically. So something I want to bring up in the context of food allergies is the idea of our bodily barriers. If someone is sensitized to a food allergen, the way we have an allergic reaction is obviously that allergen has to enter our body in a form that is able to induce an immune response. So normally our body is supposed to filter these things out so that even if you have sensitized IgE molecules, they're not going to be overreacting all the time. However, the gut lets around 2% of ingested food antigens through the barrier without sort of destroying their form to the point that they no longer cause a reaction. So that's where we can sort of get these foreign molecules into our body. And now, of course, that stacks with the fact that people who are food allergic have various immune cells in their body or immune properties that have not properly sort of passed this tolerogenic step. Normally we have T cell deletion, T cell energy, T regulatory cells that are all supposed to be keeping these things in check, so that even in that 2% case where these foreign antigens are coming into our body, those things aren't overreacting. But sort of in people with food allergies, this tends to be a sort of perfect storm of both of those going wrong. So I won't spend too much time here because if you're interested about the mechanism behind IgE-mediated food allergies, I'll have like 2 or 3 entire presentations on this because there's so much to talk about. But I'll just quickly summarize in case you're interested. But IgE-mediated food allergies are one of the ways that someone can have a food allergy. It's not the only way, but it is the most characterized, most studied, and I believe most common sort of form of food allergies. And the way it works is we obviously have these IgE molecules, which are antibodies produced by plasma cells, and they're sort of loaded onto the high affinity receptors, which are often found on mast cells and basophils. So they sort of sit there, like in this first image on the left, and when an allergen comes that is able to bind, it binds and it cross-links them, which leads to a bunch of signaling cascades, which release causes the degranulation of these mast cells, and all of those mediators in those mast cell granules are responsible for these sort of typical symptoms. Again, refer to the IgE lecture, the mast cell lecture. It has a lot on this. Something I want to talk about, though, is that you may have heard about how if you're allergic to, say, peanuts, for example, and you eat it the first time, and then you eat it a second time and a third time and a fourth time, it tends to get worse over time. And part of the reason that is because of this molecule called histamine releasing factor. Now, this is a cytokine, which what it does is it sort of primes the IgE molecules on mast cells and basophils. So a distinction to make is that there's sort of a process of activating these mast cells, but there's also a process of priming them, and priming them is typically something that makes them have an over-exaggerated response beyond what they normally have. Now let's just talk about 2 systems in allergy. So let's talk about the GI system first. Now this is responsible for a lot of, unsurprisingly, the abdominal symptoms such as pain, nausea, diarrhea, cramps, vomiting. And we see a lot of increases in gastric acid secretion. And you might be able to connect that from our histamine and antihistamine lecture, because histamine acts on H2 receptors in the gut to cause gastric acid release. We also see that there are lower levels of salt reabsorption, and this means that water is going to follow those salts out of the body, and that causes diarrhea, dehydration, which is especially dangerous in the context of anaphylactic shock. Now talking about the cutaneous system, or the skin, this is where two of the other big symptoms come in, which is urticaria and angioedema. Urticaria being hives, angioedema being more of the swelling. So these are driven by Langerhans cells, or these sort of antigen-presenting cells on our skin that also have the high affinity receptor, and these are really going to be doing a lot of work to present allergens to Th2 cells. You can see in this image over here, the reason I'm talking about the cutaneous system, and then I just talked about the GI system, is that there's something we call the gut-skin axis. And this basically is a way of describing how allergic symptoms are sort of perpetuated between two systems of our body. So you itch on the skin. This causes various mediator releases from mast cells, such as histamine, but it could also bring chemotaxis or chemokines. These are going to bring sort of more mast cells to the body, such as to the GI area, which increases permeability of the gut, which increases the ability of allergens to enter the body. So that's sort of a way that we can think of connecting these systems at large. And if you're wondering, well, what about the other symptoms? What about the respiratory system, circulatory system? Those ones are obviously going to be our life-threatening symptoms, and I'll save those for the anaphylaxis lecture. So let's talk a little bit about diagnosing food allergy clinically. Obviously this is not, it is not rocket science to see that if you eat a food and then shortly after that, within, 30 minutes, maybe an hour or two at most, you get all these, hallmark allergy symptoms, well, that's going to be pretty suggestive. So this is not, it doesn't take a rocket scientist to figure that out. However, there are ways that you can sort of preempt and sort of get ahead of those allergies rather than figuring it out too late. So one of the big things about the LEAP trial is we see that children, especially infants with atopic dermatitis and an egg allergy, are at a really high risk for developing a peanut allergy as well. So this is, again, one of the ways that we can prevent peanut allergies, or sort of try to get ahead of them. And finally, I want to mention, and this goes back to our eczema lecture, but if a child has eczema that is really uncontrolled, even though we've been controlling it, or we've been going through a standard treatment regimen, that might suggest that there is a food allergic basis to those skin symptoms on the long term. And this is a sort of flow chart that you can see. You don't worry about the specifics of everything, but I just want you to sort of understand that there is a sort of stepwise process. It's not just allergy tests. It's not just history taking. There's a lot more to it. So let's talk about the DBP-CFC. This stands for Double-Blinded Placebo-Controlled Food Challenge. Now, this is technically the gold standard for an allergy diagnosis. And if we just break down every word, well, double-blinded means the person eating the food doesn't know what the food is. It means the doctor doesn't know what the food is. It means they're also eating a placebo. And food challenge, right? They're being challenged by this food to see if they can tolerate it or not. So the reason for this is that it attempts to minimize the patient bias. Now, this is sort of a murky topic because it sort of, presupposes that the patient has a reason or might have an unconscious reason to, lie or be, convinced otherwise about the test. So, that's sort of the first thing that I think is receiving a little pushback nowadays, because a lot of times in clinic, not only are these unpractical, but we just don't really see the need to do this level of sort of control. It tends to offer pretty minimal advantage to have both the doctor and the person blind, and it can also be really scary to potentially eat a food that you don't know if you're allergic to. So most often these are either unblinded entirely, or maybe sometimes they might be single-blinded. The final thing I want to talk about, this is sort of what we all know, are these SPTs, or the skin prick tests. This is where you poke the skin with a sort of glycerinated food extract, and you compare it to a positive histamine control, which is always going to cause the wheel and flare reaction, and a negative saline control, which shouldn't. So this is really useful in excluding potential allergies. And you see this mostly with environmental and food allergens, and not as much with drugs. Intradermal tests, they go a layer deeper. These are a little more sensitive, and they are mostly used for toxins, venoms, and drugs like penicillin. And then we can also do serum tests. So these are more where they take your blood. These are very quantitative. They're not as useful to diagnose, but they're actually quite useful to predict reactivity and to trend someone's IgE levels over time, which can be suggestive that someone's outgrowing because they're getting less and less reactive. So with that is a sort of really quick overview of some points of interest in food allergy. And in the next section, we'll be talking about drug allergies.